Yeah, hi everybody. Thanks for the introduction, Marcy. My name is Kevin. I'm from the Digital Lab. I'm really excited to present at Tools and Tech about my tool called MSBooster, which is a tool for improved peptide identification and mass spectrometry-based proteomics. So as an overview, first we'll talk about why you might be interested in LC-MS, MS-based proteomics, or tandem liquid chromatography mass spectrometry-based proteomics. I'll talk about kind of what a typical experiment looks like, as well as what data analysis with our computational suite pipe will look like. And then I'll get into the specifics of my specific tool, MSBooster, in terms of the algorithm, one really exemplary use case of it, as well as some cool new features such as choosing your own models, diagnostic QC plots, and then how to use it in the graphical user interface or on the command line. And I'll round out with some future directions. So why use LC-MS and proteomics? I'm actually kind of curious if anybody here has used proteomics before, or like, yeah, thank you. Or just like worked with the data before. I know it's like a lot less of a used omics technology, but I think it's a really cool piece of, I guess, technology to work with because it provides a complementary view to other omics technologies. So I know many people are familiar with genomics or transcriptomics, but there's just so many levels of regulation in between those levels, and then finally like the proteoform that you see. And so I think by doing all those omics together in a really truly like multi-omics way, you can get a more comprehensive picture of the data that you're looking with. Furthermore, it's really cool for detecting and quantifying proteoforms, like you mentioned. So if you have these alternatively spliced isoforms, like are those actually expressed in the end? It's a good way of finding that. If you're interested in post-translational modifications like phosphorylation, the chemical modifications that will actually modify the shape and the function of your proteins, this is a really good way of doing it. And furthermore, it's a very high throughput technology where recently people have been sequencing thousands of proteins in the order of minutes to hours. And so it can really give you high throughput if you're looking at these large cohorts of many thousands of samples, perhaps. And it's just a PubMed search I did for proteomics mass spectrometry. I always like doing this when I do a talk, just to see how the state of proteomics is. And in the year 2022, 4,600 articles were published using this technology. So I think it has a lot of utility, and people are kind of valuing it more and more as it becomes more accessible. So what does a bottom-up proteomics workflow look like? Well, we call it bottom-up because we basically take our proteins and then we digest them down into individual peptide, because it's a little easier for us to sequence them that way. So first, like I mentioned, you can use an enzyme to digest your proteins. So for example, if you use a trypsin enzyme, you end up with peptides that are ending in arginines and lysines, which help us better identify kind of what peptides it might be identifying rather than looking at all possible subsequences of the protein database that you're working with. Another way of doing it is you can also directly extract endogenous peptides. So one example I'll talk about is in immunopeptidomics. You're looking at peptides that are presented naturally by your cells to the T cells. And so you can extract those as well if you just want to skip that enzymatic digestion step. Then you can do a depletion or enrichment step, such as if you're doing phosphoproteomics, you can get those phosphopeptides specifically. And then finally, this is a really important part, just you can separate all your peptides with liquid chromatography columns. That's the LC step and LC-MS. And the time at which your peptide is eluting out of the column, let me show here. Yeah, it flows through the peptide or the LC column right here. And they're separated based on different physical properties based on the peptide, such as their hydrophobicity. And when they elute out of the column, it's called the retention time, or RT. And then they can be detected in the mass spectrometer kind of across time, where each of these individual kind of groups will be a separate peptide. After you do that, and you get one of your peptide precursors entering the mass spec, you actually have two different mass spec steps, hence the MS-MS. So in the first mass spectrum scan, you can detect the mass to charge ratio of the peptide precursor. We just take the peptide mass divided by like the positive charge, for example, to get that. And then that actually is not enough to identify what peptide you have, because if you imagine you have one peptide sequence and you shuffle the amino acids around, you end up with the same mass of the peptide, but those are fundamentally two different sequences. So you actually have to do a second mass spec step in order to identify which one of those is going to be. And so to do that, we have this fragmentation step right here. So as you can see, you take your precursor that you identify in the first mass spec scan, put it into this collision cell, where you will basically throw energy at all the peptide bonds and then fragment them into these individual amino acids, these individual fragments here. And so you can see that they're being broken at every single amino acid bond. And all the fragments that are starting from the left-hand side are called p ions. And if they're from the right-hand side, they're called y ions. And then you can then shuttle all those fragments into your MS scan, where now each fragment's at mass over charge ratio and intensity is recorded in an MS spectrum. So now that we have these MS scans right here, we have to do, I guess, like the search again and see what peptide is making that spectrum. So the way we do that is by doing a database search with a database search tool such as MSBooster, which is in our pipeline. And the way that works is you can give it a protein sequence database, such as from the human protein sequence database, and then you can do this in silico digestion, where you'll take your protein sequence and then basically cut it after all those lysines and arginines if you're using like a trypsin enzyme. And then you can try to see which one of those peptides will best fit your experimental spectrum. So because you know the precursor M over Z that generated this experimental spectrum, you can narrow your list down of all the possible peptides in your peptide sequence database now to this list of potential candidates. And then we have to basically rank them somehow. And so we have the search score idea where you can basically see how many peaks are matching in between your experimental spectrum and your theoretical one. And that score is based on basically how many matches there are, where the more matches you have, the better the match, the more likely that peptide is. And then you can do that for all your peptides and then rank them in the list. And whatever has like the highest score is hypothetically your best peptide spectrum match. And this is the abbreviation for that is PSM. But within, I guess, this process, you're naturally bound to get a lot of false positives because there's a lot of noise in the spectrum. It's not as pretty, for example, as we see with this theoretical spectrum where everything is kind of the same. There's no extra stuff in there. And so how do we actually control that, those false positives? Well, we do this FDR, false discovery rate control, and we estimate that using this idea of decoy peptides. And so, first of all, before we talk about decoys, we talk about target peptides, which are all those peptides that you get from doing your in silico digestion of your protein database. And so you can see up here, we have this peptide sequence, like AYT, DE, LBE, LHR. And then if you want to get a decoy counterpart of it, you basically flip that in reverse, or you can shuffle it, for example. You make sure that the arginine or lysine at the end stays the same, but then everything else kind of gets shuffled around. And then this ends up being this peptide sequence that you expect to not be actually in your sample because it's not from the actual protein. And so this has been a really cool technique people have used. Decoys help us to estimate those false positives because they have a lot of similar features as false positives in our sample. And so when we search against our protein sequence database, we're either saying this spectrum originates from a target or a decoy. And then you can learn in a post-processing tool, such as Percolator, you'll basically visualize all your targets and decoys and then try to learn this hyperplane that'll separate them as much as possible. And so in the end, you get all these target PSMs over here that are most likely true positives or more likely true positives. And then you get your decoys clustering on the other side, along with a bunch of other target PSMs that are more likely false positive because they're more similar to those decoys, which are the false ones. And then from there, you have your peptides. You can infer your protein just by saying what protein sequences are those peptides from. Let's take a look at that. Any questions so far? So now that we have our data, like how do we actually do all this data analysis? Well, I'll talk about ProgPipe specifically, which is from our lab. It's this comprehensive proteomics data analysis suite that basically has all the tools you might possibly need. And so you can see here, there's a lot of tabs at the top that are just different steps of the pipeline. You can upload all your MZML files here, which are just the files from the mass spectrometer that give you the spectrum. And then you can do your database search with MSBooster. You can get all these cool downstream analyses. So I'm just going to shout out other members of our lab here who contributed to this pipeline. So for example, if you want to visualize the actual spectra that you're getting, you can use StrikePipe-PDB, which was made by Kai Li in our lab. And then Yi Xiao from our lab also made StrikePipe-Analyst, which gives you a bunch of cool plots afterwards. So if you have a bunch of samples that you expect to cluster based on condition or whatever, then you can generate PCA plots, you can make heat maps, etc. So those are all super helpful. So another question is, where does MSBooster fall into all of this? So MSBooster was based off this idea of open science and data reuse. I'll show here, basically, one of the downfalls of database searches is that you're not taking into account kind of the differential intensities of all your fragments. So you see here, this is the acquired spectrum. You see there's a lot of variance and how these fragments are being expressed. But in the theoretical database search, you're basically thinking that all the fragments are the same intensity, and that's truly not the case. And so what people have done in the last couple of years is they've been sharing all their mass spectrometry data online for reuse, for people to use. They can search it again. They can do whatever they want with it. And I think this has been super helpful because this data that they're depositing online, we have like millions and millions of spectra. And now we can train these machine and deep learning models to help us predict these properties of peptides. And the nice thing is that, for example, if you look at these NSM spectra, they're very reproducible across different labs or instruments. I mean, there's variation, obviously, but there's enough similarities between many different conditions that you can actually train machine learning models to predict these. And similarly for RT or retention time, the hydrophobicity of the peptide is not going to change drastically based on the LC column. And so you can also replicate that through prediction models. And so these are kind of like the foundations of what MSBoosters rely on. They use these predicted values of a peptide and compare them versus the experimental ones and say, a true positive should truly, have the predictions match the experimental values. So that's what we look for. So the MSBooster algorithm, the paper is online as of July of last year if you're interested. But the workflow of MSBooster is, first we'll go over kind of what it looks like without MSBooster. So first in FragPipe, we have your mass spec data that you're searching, and you can pass it to MSBooster for the database search. And MSBooster will give you all these features that you can then give to a tool called Percolator, which will learn the hydroplane separate between the true and false positives. And then you can do all your downstream FDR control stuff with Philosopher. But if you're using MSBooster, you want to basically propagate these new deep learning-based features to Percolator as well. So what it does is it extracts all your peptides that MSBooster gives to you, all the possible candidates, whether those are true or false or likely or not. It'll give these peptides it will format them for the appropriate deep learning model to be predicted. And then you'll get all your MS2 spectra and retention time predictions. There's also this idea of ion predictions or ion mobility, which is another feature that people have been using to separate peptides. However, we found that that's not super helpful in this pipeline. And so I'll exclude that conversation for now, but I do discuss in the paper a lot. And then once you have your predictions, you can basically, again, compare those versus your experimental ones from your mass spec data, calculate these features, and then add them to Percolator for better rescoring. What does that look like? So the input in MSBooster is these tabular pin files. I just visualize them in Excel because they're just TSV files essentially. And so every single row here is going to be, it's a little small, but every single row here is going to be a different scan, along with all the different features that describe it. You have at the end the peptide sequence, as well as the protein that it was originating from. And then MSBooster just appending these new features based on deep learning at the end. So pin files are kind of a very popular feature format because not only are they human readable, they are used by Percolator and other tools like MochaPod, which is an extension of Percolator that gives you more deep learning models or machine learning models to use. So that's what we use for processing these pin files. And in the future, there's also these XML-based PepXML files that people often use. And I haven't fully supported that yet, but that's also in the works if people want to use that for their pipelines. So the two features that MSBooster calculates mostly, the first one is this spectral similarity. And to calculate this, it's pretty simple. If you just have two vectors of scalar values, you can compare them with something like cosine similarity, for example. The way that works is you'll get your prediction. You'll say, I'll take the top 20 highest predicted intensity fragments from the prediction, and then compare that to the experimental spectrum. Are those predicted fragments actually in the observed spectrum? And if they are, do they kind of match the ratios of the intensities that we expect from the predictions? And so by default, what we use here to do that similarity calculation is called spectral entropy score. And we found that this is super helpful because it actually outperforms more traditional metrics such as cosine similarity. But if you are interested, we also do provide dot products and other things as well, if you want to mess around with that. And like I mentioned before, because it's fully integrated, MSBooster is fully integrated into BagPipe, you can use the other tools that are available. So like BagPipe PDV, you can now compare your experimental spectrum versus your predicted ones and see, is this truly a good match? So on the left-hand side here, we have this one peptide, where the top part is experimental, bottom is predicted, and the MS2 similarity between them is 0.98. This is on a scale from 0 to 1, where 1 is more similar. So it being 0.98 means that it's very likely to be true. We see that a lot of the fragments are matching. You get this Y10 ion is very intense in both the prediction and the experimental. So we'll be more confident that this is a true positive. On the other side, though, you see the experimental and predicted spectra have a similarity of 0.1. And it's very evident here. You see in the experimental, you get a lot of these B ions matching. However, it's missing a lot of these Y ions, which are quite intense in their prediction. And so because of that, we believe that this is more likely a false positive. And so the peptide is probably not going to be in our sample. The second feature we use is based on the retention time, and we calculate this retention time difference. And so basically we have to generate this locally weighted regression curve whenever we do this retention time difference calculation. That's because LC columns differ naturally. Again, they can be eluting peptides out for just like on a minute scale or for hours, and there's just a lot of differences between them. However, the general idea of what peptides should be exiting the column in the beginning versus what are exiting at the end should be pretty similar. And so we just have to calibrate between those two different scales. So to do that, we take our top 5,000 PSMs based on what has the best database search score, because we believe that these peptides already have a good database search score, should more likely to be a true positive. And so we can learn this regression curve. As you can see here, the fit is pretty good, where every dot is single PSM, and the red line is the curve that we've learned. And the idea is that basically you want a PSM to be close to this curve, because that means the difference is very small. But as you venture more and more out into the sides here, I mean, that just means that experimentally, for example, you see that like if the PSM was here, that means it's eluting at the end of the column, but it's predicted to elute at the end, then that means like it's probably not going to be true. So this is also a super helpful metric to add to the 10 files. And this is a QC plot that is always generated by MSBooster if you're using these RT features, and so you can examine that yourself as well. One caveat of this, though, is that we always have to take deep learning with a grain of salt. Sometimes you have a PTM, for example, that has never been encountered by your deep learning model before, and so its predictions aren't going to be optimal. In this case here, we see that we have a PTM of 42 Daltons, which is N-terminal acetylation, which this one model is not able to predict. And so you see that it's represented by all these black diamonds here. And that band is now kind of straying away from the main band and exploring its own thing. And so if we only generated one calibration curve, then all these N-terminally acetylated peptides would be penalized, when in fact, maybe you want to keep them because maybe they're truly important to you. In this case, N-terminal acetylation isn't really important, but the idea is generalizable. So what we do here is we provide this feature or the parameter you can called RT mass for calibration. So you can just put in the mass of your PTM that you're interested in, that you see this shift be generated for, and then it'll learn a separate calibration curve for it. So it's not heavily as penalized. But in the future, it can be a little tedious to try to find all these mass shifts yourself, and if they have to have a separate calibration curve. So I'm going to be working on automatically detecting if a mass is associated with different retention time, and then I'll generate a calibration curve automatically. So I want to give one kind of use case for MSBooster, which is HLA immunopeptidomics. There's a lot of other use cases, but I think this is a really enticing one. So HLA peptides are just those peptides that are presented on your cells to T cells for kind of like the immune response. And so if the T cell recognizes that this is like a foreign antigen, such as from a bacteria, then this immune response will happen. And so people are like really interested in identifying these peptides in order to design therapeutics. So how does MSBooster help with that? How do we help find more HLA peptides? So first, we look at this original row right here. I basically ran the simulation or this analysis 10 times, and I saw that without MSBooster, we're getting about 7,500 peptides in this data set. But now if you use this retention time feature, you get about 17% more identifications. If you use the spectral feature, you get 20% more. And if you use both of them in combination, those are two kind of orthogonal pieces of information. And so altogether, they give you about a 31% boost in the data set, which is kind of astounding. If you look at the peptides that are identified in this Venn diagram, you can see that a large portion of them are shared, about 7,000. You get this huge chunk of new peptides identified by MSBooster, but also the smaller subset of those peptides in the original set that you no longer get when using MSBooster. So I want to make sure that the peptides that we're getting are actually true positives. And so one way of doing that, thankfully in HLA peptidomics, these HLA peptides are presented on these MHC molecules, which each have their own binding specificity. And we know that in this data set, we have this monoallelic MHC that only likes to bind peptides that have this sequence right here, where it's binding leucine and methionine at position 2, and isoleucine, leucine, and valine at position 9. And so hypothetically, we'd want all these peptides from MSBooster to be, following that sequence motif. And when we do this good clustering of it, we do see, in fact, that that is the case, that a lot of these new MSBooster identified peptides follow that motif. Another way of looking at it is based on predicted binding specificity. And so there's some tools out there called NetMHC that can help you predict based on this allele, this MHC allele, how strongly it's going to bind different peptides. And so we break up, again, this Venn diagram set into MSBooster only, into the shared ones, and original only. And we see that while the peptides that are identified by MSBooster, or those shared between the two sets, are likely to be high or weak binders, that percentage decreases a lot in the original set. So more than 50% of them are predicted to be non-binders. And so those are likely to be false positives that we've ended up kicking out because we used MSBooster. And so this tool really helps you not only find more true positives, but decreasing the amount of false positives you have. One really cool thing is that MSBooster actually works better for the hard problems that database search tools do not work well for. So I've already mentioned in HLA peptidomics, we get this 30% boost. And the reason for that is this hugely expanded search space, where now peptides presented in this data set are not digested by trypsin. So you don't expect lysine and arginine at the end of all of them. They could really be any subsequence that you find in protein. And so because of that lack of constraint, then you get a lot more peptides you have to consider. And so database search tools will struggle with that much larger database. Likewise, you can see with data independent acquisition, or DIA, this is a method of sequencing your proteins or peptides where you generate a lot of these chimeric spectra, where there's going to be a lot of peptides fragmenting in the same MSMS scan. So that also presents a difficulty because now decoys have like a better chance of just like spuriously matching random fragments. So that makes it difficult for them. Single cell proteomics, you have about like a 10-50% boost. And then at the very bottom, when you're just doing a typical data dependent acquisition with trypsin, no weird PTMs out there, you see that you get like less than a 5% boost with MSBooster. And the reason for this is that this is kind of the bread and butter of database search tools. They're already like so good and so well tuned to these sort of searches that the features that are being generated by MSBooster, for instance, are already helping us identify most of the peptides there. And so adding deep learning on top of that isn't really necessary, which is kind of a cool thing to think about, that sometimes deep learning doesn't really help because you've already done all this feature engineering yourself. Any questions?
Yeah. This is a really cool application. I'm curious if there's more like spike in or more like controlled experimental data that you could use to benchmark the quality of your predictions?
Yeah, I was like, so what we know, like the ground truth of what peptides? Yeah, there's
Yeah.
actually a good amount of them there. I think people have it's like one example of that would be like a spike in E. coli sample into like a human proteome. And so I can help you basically, yeah, what is it? Yeah, you can then identify what human peptides you're identifying and what E. coli peptides you're identifying. And then like downstream, you can see like how much quantification you're getting in each of those, because you can do like an unknown quantity. And so you can like truly see like, are you identifying the peptides that you know should be in there, and like the quantity is matching what you expect. So that's one way of doing it. There's a lot of examples of that. There's also this idea of an entrapment search where you can just take your human sample, but then search it against both human and E. coli database together, for example. And you know that there should be no E. coli proteins in there. And so if you do start matching to more E. coli, then that is the case for you matching more false positives. So I did some of those entrapment searches before, and there's nothing, MSBooster did not increase the number of false positives in that case. So we're pretty confident that that is one way, that is like, MSBooster is like giving us the true positives there. That makes sense. Yeah, so that's kind of like the bulk of what that first paper was about. But I think there's a lot of cool applications for MSBooster besides that. So one thing that I'm working on right now, and the manuscript is in progress, is this idea of model selection. So in version 1.1 of our tool, we only use this tool called Diane for our spectral and RT predictions. But there's some drawbacks to Diane because it wasn't really meant to be like a prediction tool. It's used for different applications truly, and the prediction module was just one part of that. And so because of that, you know, it's not optimized for this sort of thing. So for example, I mean, first of all, it's closed source. I don't really know what the training data was, the architecture of the model. It's a little, you know, confusing there. And furthermore, Diane itself does not predict these YB1 and 2 ions, which are just if you fragment your peptide at either the left or right side, and then you just take either 1 or 2 amino acids off. Diane is not predicting that. And these fragments are actually super helpful for identifications. And so while Diane itself is doing quite well with its predictions, there's a reason that you might want to look at other models as well. And so a lot of groups have been interested in publishing more open source models for a lot of different use cases, such as chemical tags, such as different PTMs. And so I want to allow MSBooster to be able to work with all sorts of these models. And so I'll give first 2 use cases here, like again, to prove that Diane isn't always going to be optimal. So this is again with the HLA data. It's a different data set, but same idea. So we see here that Diane was able to get, how many is that? Like 15,000 peptides maybe using its features. But then if you swap in a different model, such as this PROSIT model over here, well, PROSIT was actually trained on a lot more HLA peptides, and they talked about the whole data set that they used. They did a bunch of like hundreds of thousands of synthetic HLA peptides that they sequenced and trained the model on. So because of that better data set, you get about 1,000 more peptides from this model. And so that's one use case. There's also this timsTOF instrument right here, where before I've been talking about Thermo Fisher instruments, but this is a different instrument from Bruker. And again, you see here that Diane is predicting a certain amount, but then by using a more optimal model like AlphaPept that was trained on timsTOF data specifically, again, you see an increase in peptide IDs. So already the 2 good use cases for why you want to swap in different models. And so if you're using it in the command line, you can call these different models and, you know, future release a FragPipe with the current MSBooster model with Spectra model parameter or the RT model parameter. And when we incorporate in FragPipe, we're envisioning like a drop-down menu that can kind of just, you know, plug and chug whatever model you want to do.
Would you be worried about some of these models having more false positives than others? If you're just detecting more, I mean, you could just enumerate them and end up with lots more, but that seems like that would not be a good predictor.
Right, that's true. And so I think in the manuscript, I'll try to incorporate some tests to make sure that we're getting like all the true positives and not increase false positives, like the use case I showed with like the sequence motifs and then the binding that's been predicted. That could be nice. But I think, you know, even the baseline when you're looking at the spectra itself, those intensities are just matching a lot better. You can like visually see that. So I think false positives are always, you know, an issue in proteomics. And I think we do our best to control for that. We do at, both the peptide level and the protein level. But yeah, I mean, hopefully the idea is generalizable, and by showing like a single example or maybe multiple examples in the paper, people will have some more utility in that. But I mean, it's a good point. Maybe I need to think of some way to have like a QC plot to make sure that false positives aren't increasing, for example. And I'll actually have like a slide about that kind of in the future. Yeah, Chrissy.
While you are trying to control false positive discoveries,
Mhm.
have you also made some efforts to also control for false negatives?
False negatives. Yeah, so I guess with the idea behind MSBooster is that it'll also try to save or rescue these PSMs that originally weren't good enough. So for example, if its database search score was pretty poor because maybe like not all the fragments were there or something, then initially maybe you weren't capturing that. But then with MSBooster, you see that even with the fragments that it is expressing, those intensity ratios are quite accurate, then maybe you'll save that. And so I haven't shown any plots in terms of the number of proteins they're identifying, but we do get a lot more proteins as well. So yes, it is rescuing these false negatives as well. But that being said, you don't want to go on like this, what is it called, like a wild goose chase. Sometimes the spectra itself is just so messy, it's hard to actually see that any peptide generated anything in there. And so we don't try to rescue those because, I mean, those are unsavable.
So here you compared each model against each other model.
Yes.
Is it possible that if there's a way to aggregate the model results, you might end up with a better weight, like an ensemble model?
Yeah, just like by running all the models together and then putting all those features in there. Yeah, that's a good point. That is true. I have not looked into that specifically. There is one potential downside, where if, because these models are predicting the same thing, and so they're often gonna be very linearly correlated. And we found that when you add all those features into like the pin file for Percolator itself is this linear SVM, and it actually struggles a lot with having these linearly correlated features, where now like it will tell you before the weights of the importance of each of these features. But now if you have all these different features that are telling us very similar things, then those weights start to become weird. Like maybe you'll get one. So typically you get a positive weight for this MS2 similarity score. But we've seen that, at least in the case of adding like cosine similarity and spectral entropy and like pure correlation together, those are all correlated. And when you add those all together, then you start to get like negative weights for some of them, which starts to not make sense. And you don't actually fundamentally get any like statistically significant boost of peptides after that. This might be a little different just because it's not just like different metrics you're computing for the same two spectra. You actually are using different predicted spectra to calculate them. So you actually might get a better boost, which is something I should definitely look into. But I haven't at this point. That's a good idea.
Now that you have these open data model trained, do you see that there's a dependence on the actual experimental platform that's the data was generated? So if you're testing it on one platform, but the data is collected on a different platform, that might be not as good as the same platform?
Yeah, definitely. I don't think I included anything here, but like, for example, if you have different instruments that you're doing, sometimes those spectra will differ greatly. And you'll actually be able to see it when you actually like look at, like a PSM that has really good database search score, but then no longer like those intensities don't match up anymore. So it actually looks really poor. And so that's why I have a lot of QC plots that are generated to show you, is your model acting suboptimally, right? On a more fundamental scale, how do you like correct for that? I mean, do you need to train a model that will be optimized for every single like instrument or PTM or something? That might be something that we have to do, but it's also like a piece of meta information that you can include in models to help you kind of like modulate your predictions a little bit. But that is a good point. Like sometimes you get suboptimal things from there's just a lot of parameters to calibrate with these things. So it's not always going to be good. But even that being said, we've seen that sometimes we've done predictions for these TMT tags, which change the spectra a lot. And while you see then that the predictions are suboptimal, it's still like kind of good enough that you get like a smaller boost in there, which again brings up the question of false positives. But yeah, you have to optimize things, but even if they're suboptimal, they provide some benefit. So the question is, how do we support all these different models that I just talked about? Well, Diane is just like the C++ tool that we've incorporated into our FracBite package. But all these other ones, I don't really want to go through the effort of having to, generate all these different Python environments or content environments for all of them, package all these Python tools with it. It's a lot of work because these models are being generated all the time, and that's just more work for me. And so our collaborators over in Germany have generated this Koine server. And that's super nice because they have this Triton inference server that gives you access to a lot of NVIDIA GPUs. And so the idea is that even if you're working at an institution that doesn't have access to GPUs, if all you have access to is like a laptop, for example, all you have to do is just send some curl HTTP requests over to the server, and then you'll get all of your deep learning enabled predictions, which I think is super cool because now GPU power predictions are available for everybody, hypothetically. In terms of runtime, I did it first for these retention time models. On the x-axis here, you see the number of peptides you're predicting, but I just did it for a bunch of different data sets. So here you get about 600,000 peptides, and you can predict all those in like the order of 10 to 100 seconds. So that's pretty reasonable. For the MS2 models, they take more time because now you're not just predicting a single scalar value, you're predicting that intensity for every single fragment. So it does take longer, but it's still on the order of like hundreds of seconds for the very worst model, which is AlphaPep, which is just slower than everything else. But that being said, this prediction step only needs to be done once, and then it saves it into a file like an MGF file for you to parse through and load again. So if you want to tweak with the MSBooster parameters after you've done your first search, you can just reuse those predictions. And ultimately, the prediction step is kind of out of my control. I'm not the one designing the models. It's up to the people who actually make them. And so if you kind of look at these different data sets I'm showing here, these different boxes are colored based on the step in the algorithm that is being timed. So red is when we generate the input file for the models. Green is the prediction step. Orange is when we calculate the features, and blue is for percolator post-processing. And so you see that the majority of the time here is based on the prediction itself and on the post-processing. And MSBooster itself is quite fast. And this is just from the initial publication. And since then, I've multi-threaded a lot of the steps, so it goes a lot faster. And you can, it's best if you do it on a server. We have a lot of memory and RAM. But for most analyses, if you just do it on like a desktop, I often just test on my desktop with 14 gigs of RAM. It works just fine. So now the diagnostic plots I've been mentioning. So again, you want to make sure that your model is predicting the right thing and that your data doesn't look weird. And so you provide these two different histograms. And so in blue here, you basically see all your target PSMs, and in orange, you see your decoy ones. And from your target ones, you expect to see this bimodal distribution of your scores, where this is going to be your M2 similarity and this is RT difference. But you expect this bimodal distribution because you want your true positives in one distribution and then your false positives in the other. And you also want your false positive targets to be matching the decoy PSMs. And so in this case, we see that here we see like this bump is being matched quite well on both targets and decoys. Same thing in the RT difference. And so when you're looking at these plots for the MS2 similarity, the more on the right you are, the higher the similarity you have, which is more likely to be true positive. And for the RT difference, you want that difference to be small, and so you want to be on the more on the left-hand side. But sometimes things do go wrong. And so there's two cases right here that I'm showing, where now in this file, we see that you get a lot of decoys and false positives, and you still see the small bump in your true positive targets, but it's much smaller. So this is not necessarily an issue in and of itself. It just means that in your data set, perhaps you have just not a lot of targets there. If you do a lot of fractionation of your data, for example, maybe you just get the final couple of peptides eluting at the end. Another possibility for this is maybe you use the wrong database. So maybe you have your human data set and you have like a very different species that you're matching to by accident because you forgot to change your database. Well, now you only get like a few of these true positive because you're only maybe getting a few of those homologous sequences. So that's one thing that you could learn from your histograms. Another thing here is, like I mentioned before, with not having the optimal prediction model. In this case, you see that while you do still see this shift toward the right-hand side of your peptides kind of being similar, there's also just not this like huge concentration around one. And so in this case, we're using a model that is not truly optimized for your data set. And while it'll still be helpful, it's definitely not as good as it could be. Like I mentioned before for Koina, this is just a model or the figure that will show you on the x-axis, the timing, and on the right-hand side, the y-axis, you'll see the number of peptides that have been predicted in thousands. So if you just want to see how quickly it's being done, you can see it in this figure. And then this is an interesting plot right here, is this normalized collision energy calibration plot, which again, is currently in the manuscript is in progress, but the idea is interesting, where at this step, when you're putting energy into the peptide and fragmenting them, you can actually customize how much energy you put into that step. And if you put in less energy, then fewer of those bonds are fragmented, which could be helpful for something like a label modification, like phosphorylation, where those tend to fall very easily. And you want to use like less energy in order to keep that phosphorylation on the peptides that can localize it. And then you might also want to use more energy, for example, if you want better fragmentation. And so this collision energy parameter has a lot of effect on the actual peptides predictions. And so in order to calibrate that, these values differ a lot between instruments. So like saying 30% on one instrument isn't the same as on a different instrument. And so what we do here is we take the top 1,000 PSMs, again, ranked by their database search score, give it a Koina, and then predicts it at this whole range of MCE values. And then we take the highest median similarity across the NCEs, and then choose that as the optimal collision energy for all predictions in the future. So one thing is also that the box and Wishkal plots here are just super chunky. So Java doesn't really have a lot of good, graphing packages, unfortunately, such as like Python or R. And so I messed around with this a lot, but it doesn't really look that good, but you can kind of still generally get the idea behind it. So how do you use MSBooster and FragPipe? Well, all our code is open source, minus a few things like MSFragger is a little more closed source, but a lot of our tools are very open source. And so you can see how we're doing everything. You can provide your own commits to our repositories, and we can pull them if they're good. It's a very popular tool. So MSBooster has been is packaged now with FragPipe, and FragPipe has been downloaded 47,000 times across all 21 versions. So we're always coming up with improvements to the tools. And yeah, we have a very active presence on GitHub, always solving different issues that people have, addressing different feature requests. So if you do end up using Proteomics for your data analysis and want to use FragPipe, we can definitely help you out there and answer your questions. Or if you have any weird chemical tags that you're synthesizing for your proteins, we can also help adapt our pipeline for them. That being said, the FragPipe GUI has been very popular. It's been very popularly used. However, the command line version of MSBooster is much less used. I'm mostly the one who's using it, but there's a couple other people maybe. But if you're interested in using the command line version, which has a lot more parameters for you to mess around with, I actually just updated the GitHub documentation today. So I have a lot more details about how you can do that. And if you have any questions, you can just contact me as well. Like I mentioned, MSBooster and FragPipe, very easy to use. Currently, in the current version, all you can do is basically click a checkbox and say, I want to run MSBooster. And then you can say, I want to use RT features or Spectra features. You can turn those on or off. There's also this one called use correlated features, which I'm not a particular fan of, but kind of addresses the point that we made before about you can have your pin file now having both spectral entropy and potent similarity and all these correlated features. It's not super helpful, but maybe you can dig around with it, and maybe there's a use case for it. If you want to use MSBooster via the command line, well, it's supported on Windows or Linux, and if you want to run it, it's a Java-based tool. So you can just do Java jar, provide a path to the jar file for MSBooster, and then you have this parameter list. And this is where you provide all your parameters. At the very minimum, all you need to provide is the location of your Diane executable file for the prediction model. You provided a path to your MCML directory that has your mass spec data, and then you can give it the pin pepxml directory, which is where all your pin files are as well. But there's other parameters that are also accepted and are documented on GitHub. And for future directions, well, I've spent the last year and a half really just thinking of more and more really cool ideas, and I'm presenting at these conferences and not publishing yet because I'm very lazy about writing. But I have all these things on the docket here that I think will be super helpful to people. So one idea is that you can use these models that predict non-YP fragments, because everything I've talked up to this point about is only predicting Y or B. But there's also different fragmentation modes that you might be interested in. Also, in the case of HLA peptides, you get a lot of these internal fragments where you get fragmentation both on the left and right-hand side. So you get these like internal sequences, but those aren't predicted by current models. And so I'm going to incorporate some of those to help improve rescoring again. If you want to set up your own local Kona server, you can do that with a Docker image that the Kona team provided. This might be nice if you want more privacy for your data. If you have lab-specific models that you're training that you want to host on your own server, then you can do that. And then finally, you have transfer learning with AlphaPeptide. And transfer learning, I know some of you are familiar with, but it's this idea of if you want to specialize your model for your own use case. So we already have this baseline AlphaPeptide model, which is really powerful. But if, for example, you want to train like a phosphorylation specific model, and AlphaPeptide is not doing as well as that, as well on the data set, well, now I can just give the model maybe like a few thousand PSMs that have your data, that have your phosphorylation peptides in it, and then it can learn those specific phosphopeptide patterns. And then just by using a small subset of your data, you can train a very powerful model for your specific use case. So truly, I'm just envisioning MSBooster to be kind of like this playground. We can mess around with models and see like what works for you, and then help you train new models. So this is kind of what I'm going to be implementing in the next year or so. So be on the lookout for that, and yeah, just see all the cool features I'll be adding. Yeah, so here's my contact information. If you want to get in touch with me, we have a lab page along with the FragPipe page that will link you to GitHub. If you want to use the QR codes, you can go to FragPipe or you can read my manuscript. And yeah, there's some pictures of our lab. We don't actually have any current photos that have everybody in there. The lab is currently changing a lot. Like Jenny, you're in the lab, you're not in these pictures. But yeah, no, we're all like really fun people. We're all very helpful. And again, we're always excited to work with new collaborators and answer questions. So if you want to reach out to us and talk about your specific use case or anything, yeah, please reach out to us.
Any questions? If you're online, you have a question, you can put it in the Q&A box. Are there any questions in the room?
Yeah. You mentioned the DIAA-NN model to start was closed and you just used it as is, and then you said you switched to these open models.
Yeah.
And then you talked about fine tuning your specific set, but I missed at some point, like, how specific are these models for the specific application? Like, what are they
Yeah.
trained for, and is there a mismatch between what they're trained for and your application?
What Diana's trained for, or just generally?
I mean, like the newer ones that you.
Oh, yeah. So they try to be as diverse as possible, and a lot of them are publishing their training data sets along with it. So they will, I mean, the really cool use case, I think, is the Prosa team, because they have this wet lab that generates a lot of synthetic peptides, like millions of them, and those are all available online. And those span a wide range of normalized collision energies. They span different instruments. They have those triptychs data sets, non-triptych data sets, a lot of PTMs that they're looking at specifically, like they synthesized 21 different PTMs. And so the idea is that they're as generalizable as possible. So hopefully,
They have like gold, they like know what the peptide is and know what the spectra is,
Yes.
and they're trying to predict the peptides from the spectra.
exactly, yeah.
Okay.
So that data set is readily available, but there's also this idea that sometimes when you put in like so many different like PTM, for example, into a model, it like starts to like forget about the other PTM patterns, right? So I'm more of an advocate for having more specialized models when you have those available. But because there's like so many things to train on, it's impossible to cover all of them. And so I think for most use cases, and where like not all PTMs will actually change your spectra or within the time that much, I think the generalizable model that they have is actually quite good. And I'll be showing that in the manuscript as well. At least for HLA and non-triptych, they perform very well because they have huge data sets for them.
Any other questions? No, let's thank them one more time. Thank you everyone for coming, and hopefully you come next week.